I want you people involve as much as possible in the discussion, because this chapter, all the questions will be critical thinking question. You will not find any straightforward questions. So better you understand properly and keep doing mistakes while answering, so that you will not do again the same mistake when you go for the exams. Okay, so regarding the alcoholic OH, I hope all you remember it need to be above the plane in the R isomer R and enantiomer, right? Because in the R form, it forms the important hydrogen bonding. So this key information you know. And regarding the catechol group, catechol OH, how many information you are supposed to remember regarding catechol OH? Quickly. See, there are two catechol OH.
Yeah, they need 2 points. They need to be ortho.
Yeah. Very good. When the two OH group attached to benzene ring, yeah, when they are attached to the ortho group, ortho to each other, they are known as catechol. What is the importance of catechol? What is the advantage and disadvantage of catechol?
Make hydrogen bonding with serine.
Yeah. So requirement of catechol group is it is required to form important hydrogen bonding with serine 207 and 204. But this catechol group also have two disadvantages, right? What are the disadvantages? Two disadvantages.
Short duration.
Not orally available, and its duration is short.
What is the reason why? What is said is correct. Not orally available. Yeah. Why short duration?
Because it goes under two reactions, two metabolized reactions.
Yes. Yes. What are those?
Called O-methyltransferase, the COMT,
Yes.
and the sulfokinase in the wall.
Yeah. Which enzyme is responsible for what? And what is the chemical changes you see?
The duration is caused by the O-methyltransferase, and we see the methyl transferring, adding a methyl group to the OH.
What is that? Oh, another group, catechol O-methyl. You see in the name itself, it is not O-methyl, catechol O-methyl. So this enzyme COMT will act only when the OH is in the form of catechol. Okay, so it will convert the upper OH into OCH3, and this is responsible for short duration of action. Sulfokinase, which is present in the gut, it will convert the bottom OH into OSO4, right? And therefore it is orally not available. So with this understanding, we will carry forward. We will not waste much time because our class timing has been reduced. So based on this, we have seen the adrenaline. What are the disadvantages? We have seen it is not orally available. It has short duration of action, and it binds to both alpha and beta 1 because it binds to alpha 1. Okay, it binds to alpha receptor. It shows pressure effect. Because it binds to beta 1, it shows cardiovascular side effect, right? Then we have seen what is the reason it binds. The reason we have seen that it is then we discuss this concept. I go to the slide. Yeah, this concept. If the alkyl group is methyl, it can bind to all the hydrophobic pocket. Just recalling, because we have studied the hydrophobic pocket of alpha receptor is smaller than beta 1. Beta 1 is smaller than beta 1 in beta 2. So in other words, the hydrophobic pocket of beta 2 receptor is bigger than beta 1, and beta 1 is bigger than alpha. So the smallest alkyl group, that is a methyl group, it can bind to all the hydrophobic pocket, because of which it can show, if it is the methyl group, it will show the pressure effect. It will also show the cardiovascular side effect. But if it is isopropyl group, it cannot bind to the alpha. So isopropyl functional group, it will not show the pressure effect. It will only show the cardiovascular side effect. Remember this point. Today I'm going to ask a lot of questions at the end of the lecture. Okay. And if it is tertiary butyl, then that means it will bind only to beta 2. That means if the alkyl group is tertiary butyl, it will not show which type of side effect. It will not show pressure effect, and it will not show cardiovascular side effect.
Excuse me, doctor, but I don't have, sorry to interrupt, but I don't have that slide in my PDF. This one.
Yeah, these slides I prepared during the lecture, not when I gave the PDF. Okay. So when you
Okay.
go through the video recording, you will get the slides.
But can you please post updated slides?
Okay.
Or
Because
is
these
it
slides
not
are
very important?
okay. I can give it. These slides are something like my white blackboard. You know, when I'm in the class, I draw this in the blackboard.
خلاص, okay, doctor. Then we will write it as a note. It's okay then.
Okay. Anyway, I will give it. I will update it because these are like messy slide, but informative, I guess. Like for example, you see this slide is also not there in your PDF. What you have is this one. So I just cut down into this one. Easy to understand. So with this understanding, then we have studied the isoprenaline. We have seen the advantage of isoprenaline and disadvantages. So I'm not going to repeat this one. Now, based on this understanding, today we'll move on to another drug, the orsiprenaline. Okay. So look at the structure of orsiprenaline. Okay. Look at the structure of orsiprenaline. Can you tell me what are the advantages you see in orsiprenaline as compared to isoprenaline?
It has pressure effects and cardiovascular effects.
Okay, because it has the isopropyl group. No. Once again. Orsiprenaline, the first one. Can you repeat? What is the disadvantage you see as compared to isoprenaline?
Orally
I think
active.
like
Can be
will-
given orally.
No. Why?
Orally
It has a
active.
no blood-brain
Longer
barrier.
duration.
Does not penetrate the BBB.
Once again.
Not
Does
as
not BBB.
to catechol or methyl transferase.
So it is stable to COMT or not stable?
Stable.
Yes.
It is stable, right? How about the sulfokinase?
Stable to sulfur.
Stable.
Yeah, so it is stable. Orsiprenaline, it is stable to both sulfokinase and COMT. So it can be given orally. It has a longer duration of action. What is the another side effect you see in terms of selectivity?
Cardiovascular.
Cardiovascular side effects.
Cardiovascular only?
Yeah.
Why?
Only. Because it's
It's beta selective.
It is? Okay.
Very-
What do you mean by beta selective?
Isopropyl.
Pocket is big.
Yeah,
Fit
more
the
specific.
beta hydrophobic.
So it won't bind to the alpha.
Pocket.
Right.
Yes, that is better answer. It won't bind to the alpha. So when you say beta selective, you have to mention there are two beta, remember, beta 1 and beta 2. So you have to tell why it shows cardiovascular. Is it because of beta 1 or is it because of binding to beta 2?
beta 1.
Yes, it shows cardiovascular. So you see, you have to answer it in a proper manner. So you have to say, because the isopropyl group is bulky, it will not fit into the smaller hydrophobic pocket of the alpha receptor.
Yeah.
But it can fit into the hydrophobic pocket of the beta 1 receptor. By binding into the beta 1 receptor, it will show the cardiovascular side effect. So if you simply say it is beta selective, it is incomplete answer because there are two beta 1 and beta 2. So side effect is because of beta 1. So be specific while answering. Okay. So after orsiprenaline, you see there is another drug, salbutamol. Look at the structure, salbutamol. How many advantages you see in the salbutamol? Don't read. Give a chance to your mind.
we have tertiary butyl.
Okay. So.
And we have an ether, ethanol.
Mhm.
And it is in meta position.
Okay. Tell me the
Longer
advantage.
duration.
Okay.
So
Is it stable? Is it
makes it
stable to
more-
COMT?
It is stable to comet.
How about the sulfokinase?
Yeah, stable.
No, not stable.
No, not
It
stable.
is also stable to sulfokinase. You
Stable.
see, yeah, it is not a catechol system. Okay. And moreover, because of the presence of CH2OH, it will create a steric hindrance to the next OH. So sulfokinase cannot attack. Salbutamol is stable to both COMT and sulfokinase. Now, regarding the tertiary butyl, what is the advantage and disadvantage you see?
It makes it more
It's
selective for
more
beta
selective
than beta 1.
Beta. Okay, it is
for
more
beta.
selective. it will more selectively bind to beta 2. So if I ask you, what is the advantage of salbutamol as compared to what is the main advantage of salbutamol as compared to orsiprenylene, and what is the advantage of salbutamol as compared to isoprenylene?
Okay, can you repeat that, please? Can you please repeat?
Okay, I ask one by one. What is the advantage of salbutamol as compared to orsiprenylene, the first structure?
Okay. Terbutaline, it is stable to COMT and sulfokinase. Okay, as to isoprenyline, it's more selective to beta 2 than beta 1.
Okay.
Is that it?
You know the answer, but I think you are struggling to answer it correctly. See, I repeat my question. Okay, I repeat my question. Okay, I rotate the structure. My question is, what is the advantage of salbutamol as compared to orsiprenylene? Forget about isoprenylene. The advantage of salbutamol as compared to orsiprenylene.
No
Vascular side
effects.
Yes. So how many advantage you see? Only
one advantage.
One.
One advantage. So give reason. The salbutamol will not have cardiovascular side effect as compared to orsiprenylene. What is the reason?
Selective for
Terbutal.
That will only fit to the beta 2 receptor.
Yeah. So it has. Yeah. So it has a bulky tertiary butyl group which can fit only into the bigger hydrophobic pocket of beta 2 receptor. It will not fit into the smaller hydrophobic pocket of beta 1 receptor. Okay.
Yes.
Whereas in orsiprenylene, it has isopropyl group which can fit into both, you know, the smaller pocket of hydrophobic pocket of beta 1 receptor, including the bigger hydrophobic pocket of beta 2 receptor. So it will show cardiovascular side effect. Now my next question. What is the how many advantage you see in salbutamol as compared to isoprenylene?
3.
3 advantage. Very good. What are those?
The category group not present in salbutamol, so it will be available orally, and the short long duration and selectivity will increase with the salbutamol because of the tertiary butyl.
So it will not show cardiovascular side effect. So always careful. I am telling you again and again in every class. Try to answer it full. Always you, because you practice more, you will be able to do it. When you say selectivity, you tell the effect also. Okay. All right. So we go to the information of salbutamol. So I need not to teach this slide, to be honest. You already answered it. But there is only one point that I did not discuss about salbutamol. If you look at this structure, because of CH2OH, okay, the OH become more closer to serine, okay, by one bond length, as a result of which it can form stronger hydrogen bonding compared to the catechol group. Is it clear what I said?
Yeah, clear.
Yeah.
What about the another OH? It will be near to?
Yes, no, another OH it will form normally. It will form normally, but another OH that is on the top, it will be more closer because of one increase of one bond length. Okay, because CH2 group is in between, so it goes closer and forms stronger hydrogen bonding, which also increase the potency. Okay, theoretically, but initially, when it was given, when it was introduced in the market, its potency was found to be same as isoprenylene because it was given as a racemic mixture. Initially, it was administered as a racemic mixture, but in the racemic mixture, you know, which enantiomer is more active, R or S?
R.
R is more active. Okay, but if you give only R enantiomer, then definitely it is more potent as compared to the isoprenylene. All right. If I ask you what are the 2 reasons, tell me 2 reasons why salbutamol is more potent than isoprenylene. What are the 2 reasons you will give?
Because it's bound with the serine 207 stronger than the catechol, the normal catechol.
Okay. What is the second reason?
So it's more selective.
R. It's R.
Yes.
So no,
Yeah.
vascular
The alkyl
site.
group. Alkyl
Yeah,
group,
yeah.
because it is tertiary butyl, it will bind more to beta 2. Whereas in case of isoprenaline, it will bind to all beta 1 and beta 2. So you will not see 100% action. All right. Okay, so I move to the next, terbutaline. Next example is terbutaline. So if you remember these key points and keywords, you don't need to remember the whole slide. Entire chapter is based on few concepts only. So terbutaline should reveal the salbutamol, whose chemist replaced the tertiary butyl group on the N atom of the orsiprenylene. So if you look at the modification of the structure, actually terbutaline was discovered by modifying the orsiprenylene. So what is the advantage of terbutaline as compared to orsiprenylene, you say?
It's no cardiovascular side effect.
Cardiovascular side effect.
Beta 2
Okay, so because it is selective to beta 2, because of the presence of tertiary butyl group, it will not have cardiovascular side effect like orsiprenylene. All right. So by rational design process, we now have drug. Okay, we are comparing with that. We started designing and developing drug as compared to adrenaline. All right. So we have drug now that don't cause insomnia, stable to COMT, stable to SK, sulfokinase, and do not agonize the alpha receptor, and which cause the pressure effect. That means now they don't show the pressure effect, and it is more selective to beta 2 receptor. Okay, to show the desired bronchodilation, and do not bind to beta 1, which cause the cardiac side effects. Right. So then what they did, they synthesized some longer acting beta 2 agonist. So if you look at this structure, can you see a similarity in this structure, salmeterol with the salbutamol? What similarity you see in this structure? There is one thing that is similar with the salbutamol.
The catechol OH have a CH2, so it will be longer. So it will be the hydrogen bond with the serine will be stronger.
Stronger, very good. So it will show stronger hydrogen bonding as compared to the catechol, and moreover, you see it has a very long hydrophobic group. So hydrophobicity has strongly increased. Because of the increased hydrophobicity, or you can say the lipophilicity, it allows the drug to partition into lipid bilayer of the bronchial cell. So it will allow the drug to be trapped into the cell membrane bilayer. You know the cell membrane, they are hydrophobic in nature. And then once it is trapped there, it will be released from there very slowly into the aqueous biphase. Then it will agonize the beta 2 receptor. This entrapment of drug inside the bronchial cell membrane remaining for longer period, it increases the duration of the drug. How long? It increases the duration of the drug for 12 hours. So it can be given for prophylactic action, and it increase the dosage. So if it is 12-hour duration, means how many times a day you can give?
Twice daily dosing.
Twice daily. Okay, and it is found to be 2 times more active than salbutamol. And the slow release from the bilayer means it is unsuitable for acute asthma attack. It is only given for the chronic asthma patient as a prophylactic agent, right?
Doctor, can we give it orally?
Which one? Yes.
Because of the long hydrophobic chain.
Yes, and it will mainly given by inhalation. So when you give by inhalation, right? So in the lungs itself, it will get entrapped because of the high lipophilicity. It will get entrapped inside the bronchial cell membrane, and from there it will release slowly and bind with the beta 2 receptor. And it is stable to both COMT and sulfokinase. Now, same. If you look at the formoterol, what do you see in the formoterol?
Formido group.
Yeah, formido group. So because of the formido group, this formido group also further improve the hydrogen bonding with the receptor, and it is found to be 2 times more potent than the salmeterol. Right. The probability of hydrogen bonding formation here is formido group is more stronger. Okay. And as the lipophilicity is between salbutamol and salmeterol, its onset of action is only 1 to 3 minutes. Now, can I ask you one thing? If the onset of action is 1 to 3 minutes, don't you think it can be given for acute asthma attack?
Yes.
Can it be given?
Yes.
No.
No,
Yes,
it's too long.
why not? Yes. So, as a normal person, have you tried? Can you hold your breath for 1 minute?
No, will cause brain damage.
Yes, you cannot hold even as a normal person. You know, it is difficult to hold the breath for 30 seconds, even for 1 hour. Okay? So 1 to 3 minutes is still long. So still it prevents the use for asthma attack, acute asthma attack. It is given only for the chronic. Now here, next example is Benbuterol. Here I'm going to ask you a common question. Benbuterol is an orally active prodrug of Terbutaline. So if you look at the structure, Benbuterol was developed actually from the Terbutaline. So can you tell me what is the 100% advantage you see in Benbuterol as compared to Terbutaline?
The prodrug.
Yeah, it's a prodrug, and you see both the OH are fully sealed. How the both two of the phenolic groups were masked, okay, by converting them into bis bicarbonate. Bis means two bicarbonate. You see there. So when it goes inside the body, the Benbuterol will be slowly converting back into the terbutaline with the help of two enzymes which will break this carbamate bond. What are the enzymes? That is plasma cholinesterase and oxidative enzyme slowly degrade the molecule, allowing and which allows only once daily oral dosing. Now, can I ask you, if a drug has only once in a day dosing, what is the advantage you see with that drug? Any kind of, it can be respiratory drug, cardiovascular drug, anti-cancer drug, any drug. Just a
Long
common
duration.
concept. What is that? Yes.
It's a long duration.
Yeah. So what is the advantage?
The frequency decrease.
Yeah. So what is the advantage
Patient
actually?
compliance.
Yes, patient compliance. It increases the, whenever you see longer duration, less dosing means it is always linked to patient compliance. That is likelihood of the patient compliance, increases the patient compliance. Now, second question, I taught you in anti-cancer, and also I asked you that I'll ask the same question. Whenever it is a prodrug, what is the one most common disadvantage that come to your mind? I give you a clue. Which type of patient you cannot give a prodrug?
Liver failure.
Yes. So one of the common disadvantages, it cannot be given to a patient who is suffering from hepatic insufficiency. So just based on this concept, I can simply ask a question. Which of the following respiratory drug, cannot be given to a patient suffering from hepatic insufficiency? What should be your answer? Benbuterol. And in the whole chapter, how many prodrug you have studied?
One.
Only
One.
one.
Only one?
This one.
Yeah, this one, Benbuterol.
This is the first
So like
one.
that, yeah, this is the only one you are studying in this chapter. So remember, these are the specific questions I generally ask, and specific information. Wherever there is a specific information, especially whoever is attending today's class, if I ask this question, definitely you will be able to answer it. Okay. Sometimes simple concept we get confused in the exam because of stress. Now I go to the summary. Okay. So if you look at the summary of the class, so try to recall, look at the structure. Adrenaline, which is naturally available inside human body, and it was extracted from where it was initially extracted to fulfill the need of the asthma patient. Do you remember?
Yep.
From?
Sheep.
Sheep. From the adrenal gland of the sheep as a glycerol extract. So what are the points you see in adrenaline? What are the disadvantages?
Oral activity. And short duration.
Oral activity means orally available or not available?
Not available.
Not available.
Very good. And then?
And cardio side effects.
Pressure side effect.
Yeah, pressure side effect is because of what?
Alpha bind to alpha 1 receptor.
Yes, and cardiac is because of
Bind to beta 1.
CH3, the methyl group.
yes. Which functional group is responsible for this? Methyl group, right?
Methyl group.
Okay. Okay. So if you are able to understand this summary, you can answer any questions in the exam. So then we have seen the synthesized synthetic version was the racemic version, right? Racemic means it was a synthetic version was SNR, right? So which one is more potent and why?
R more potent because OH above the
Plane.
Yes, in R form, the alcohol, which OH?
alcoholic OH.
The one.
Yeah, you see in the middle, right? Side chain. So remember, this is the third time I'm mentioning. Whenever you say OH, don't simply say OH. You mention which OH, catecholic OH or alcoholic secondary OH. You need to mention. So R enantiomer R is more potent than S enantiomer R. It is because of this alcoholic OH, which remains above the plane and can form important hydrogen bonding with the amino acid residue. What is the name of the residue?
ISN 293.
Okay. So participate quickly, because at the end, after the summary, I have very good questions for you to solve, which will help you to prepare for your next quiz. So to overcome the problem of adrenaline, ephedrine was discovered. So what were the advantages and disadvantages of ephedrine?
Not possessing a catechol system.
So no catechol system. That
Less
means
potency.
yes, it is stable to. The advantage is it is stable to COMT and SK, right? That means orally available, longer lasting. And disadvantage is less potent. Why less potent?
No, the absence of a catechol.
Yeah. So,
There's not
yeah.
bind in 204 and 207.
Hydrogen bond to serine.
Okay, very good. And what was the another additional disadvantage?
Cardiovascular and pressure side effect.
Yeah,
It
that's
is a
side
common.
Common,
Cardiovascular
yes.
pressure is same as adrenaline, but what was the
Yeah.
additional disadvantage you see?
More
CNS side effects.
effects, more
Read here. Lock P.
Yeah.
Okay. Removal of OH group, introduction of methyl group, it increase the lipophilicity. So increase the lock P, which allowed the penetration to blood-brain barrier and causes insomnia side effect. So to overcome the side effects, what they did, they introduced the catechol group again and discovered isoprenaline. So what is the advantage and disadvantage of isoprenaline compared to ephedrine?
The log P.
Lock P reduced, so no insomnia, right? No blood-brain barrier penetration, no insomnia.
More potent because there is catechol OH.
Okay, more potent, very good. Then, what is another advantage?
It has the no pressure effect.
No pressure effect.
Because
Yeah, because
Ibuprofen.
selective to, yeah, but it is non-selective to
beta.
beta 1. The cardiac side effect is because of which receptor?
Beta 1.
Yeah, so cardiac side effect is because of beta 1, right? Okay. So because of the catechol, what are the other disadvantage? It is unstable to COMT and SK. Then came the orsiprenaline. If you look at the orsiprenaline, what is the advantage of orsiprenaline compared to isoprenaline?
It's stable to SK and COMT.
Yeah, stable to SK and COMT. That means longer lasting, orally available. Which one is more potent, isoprenaline or orsiprenaline?
Isoprenaline.
Why orsiprenaline is less potent?
have a catechol group, so will bind to hydrogen bond on serine.
My asking question was why orsiprenylene is less potent?
Not forming H bond with the serine 2047.
Yeah, it cannot form two hydrogen bonding with the serine because the catechols are removed apart from each other, right? Then compared to orsiprenylene, they discovered the salbutamol. So compare again salbutamol. What are the advantages you see in salbutamol?
It has hydroxymethyl, which makes it stable to the COMT and form a stronger H bond with the serine 204.
Very good. So it has more potent. And then what is another advantage you see?
Beta 2
tertiary butyl, it makes it more selective for beta 2 than beta 1.
Okay, so what happens? What is the effect you see?
No cardiovascular side effect.
No cardiovascular, and also definitely no pressure effect, because we are comparing this one with the orsiprenylene. So I have written only no cardiovascular. But if you see if you compare this with the adrenaline, no cardiovascular side effect, no pressure effect. Okay. So when you see the terbutaline, you know already. So it is stable to COMT-SK, but less potent as compared to salbutamol. Okay. You know why it is less potent?
Because lack of catechol.
Yeah, catechol. Moreover, salbutamol can form stronger hydrogen bonding, right? Because of the CH2OH. And one common advantage, that is both are beta 2 selective, no cardiovascular side effect. Then from terbutaline, you see came the salmeterol. Salmeterol has increased duration of action, but not good for the acute attack. So you remember this one. It is given only for the prophylaxis. Then eformetrol, because of the formido group, it forms better hydrogen bonding with the receptor, but still it is not good for the acute attack. Then we have seen one prodrug, that is the benbuterol. It's a prodrug of terbutaline, right? It's orally active and long duration, and it's given only once in a day. Still, it is given only for the prophylaxis, beta 2 selective, so no cardiovascular side effect. So now I go to one of the questions. Tell me the answer of this question. Read this question and tell me. Which of the following drug? Okay, see there are 4 structure given. Structure 1, 2, 3, 4. I'm going to ask 2, 3 questions out of this. Cannot be given for the treatment of asthma to a patient who is also suffering from hypertension. That means a patient has two problems, asthma and hypertension.
Okay, we can't give compound 1.
1, because it shows pressure effect.
One.
Yeah, 1.
Binding side.
Because it points to alpha and beta 1.
Only 1?
And 3.
Yes. Why 3?
It fits with the hydrophobic pocket for beta to beta 1 too.
Yes. Why? Because it has isopropyl group, correct?
Isopropyl, yes.
See, structure 3 and 1 cannot be given because 1 has methyl group which can bind to both alpha and beta 1. Structure 3, it has isopropyl group, so it can still bind to beta 1 and show cardiovascular side effect. So structure 1 and 3 cannot be given. Am I right? So
yes.
Yes, you are.
Yeah. Same question I change. All right. See the same question again. Can be given.
3 and 4. 2 and 4.
Answer is 2 and 4. Why?
Because it will not show the pressure effect as a side effect.
No. Answer is wrong.
Because they are not binding to the alpha.
Selectivity.
Yeah, hypertension. You see the question, hypertension. The hypertension is
Oh,
mainly
can be given.
yeah, can be. That's why I say you read the question. So can be means what is the reason?
The second one.
The
The second structure can be given.
Can be given. Only second? And why?
No, the fourth one.
Because it has a bulky side chain that won't bind to alpha or beta 1 and only bind to beta 2. So no cardiac side effect and no press side effect.
Very good. So mainly it will not bind to the beta 1. So what is the name of the group? You have to know the name.
Tertiary butyl.
Yeah.
Tertiary butyl group. Good. So see, I hope if you understood this, you will be able to answer many questions. Enter chapter, you will have questions like this only. Okay, next question. Which of the following structure is
For the
selective?
previous one.
Yeah.
Why we cannot give the structure number 4?
You see, same tertiary butyl.
Yeah.
So it can be given.
Yeah. Okay. Can be given.
Yeah, can.
So
you see,
2 and 4. Yes.
2 and 4 both.
Okay. Thank you.
Correct answer is 2 and 4. So now this one, can you answer me? Which of the following structure is a selective beta 2 adrenoreceptor agonist?
A.
Only A?
Yeah, only A.
Yeah, only. As far as I know.
Okay. Change my question. Now tell me.
A and B.
No, B.
A?
No, B, no. A, just A.
Think carefully. Let me see
A
who can
can't
answer.
bind to beta 1. So
A.
A is out of question, right?
A and
So what
B.
is the correct answer?
A and C.
A and B.
No. All of you wrong. What is the
I think
question?
A and C.
See,
no.
Which
read the
difference?
question. Read the question. I enlarge the question again.
He said
Which
1
of
and
the
2.
following selective beta 1 and beta 2, which will bind to
B. B. Only B.
I can write non-selective beta 1 and beta 2. That means which can bind to both beta 1 and beta 2?
D.
B.
D only.
D only? No.
E.
All except A.
Yes.
Why? It
answered
has
the question?
tertiary. Oh, it's not tertiary.
Give me a bonus, please.
See, when you are able to answer here, that means automatically bonus. You will be able to answer in the exam. You see,
Yeah.
everybody takes mistake. The same question I asked in the previous slide, right? It is the
Yes.
same question, just in a different way of asking. This is more straightforward. See, you misread. I am asking question to train your mind, okay? So which one, the question is, which one can bind to both beta 1 and beta 2?
The
So do you
smaller it
understand?
could be.
See, all C, B, C, and D, all of them can bind to beta 1 and beta 2.
Yes.
Yes, sir. Do you understand, Osama? Yes or not?
Yes, sir, I understand. It's about this, the tertiary group on the side.
So see, A cannot bind to
Yeah, it can
beta
bind to
1.
anything. That's why I said
Yeah.
it's the first thing is out of the question, but it's the matter of with the rest. Okay.
Okay.
So
you can, yeah. So methyl means it can bind to alpha, beta 1, beta 2. That means it can bind to beta 1 and beta 2, right? In structure D also it is methyl. That means it can still bind to beta 1 and beta 2 because it's the methyl. Structure B is isopropyl. Isopropyl means it can bind to still beta 1 and beta 2. So please don't misread the question when you go for the exam. I will play around with the structures. Next one. Which of the following is an end product of the action of sulfokinase on adrenaline?
A and D. No, A.
C. B. Only B.
ما في
How about
category.
the others?
B.
Okay, Basil said B. Who is another person say B?
Yeah, me. Mara.
Mara. Okay, how about the others?
B.
Osama?
I'm not sure. I'm not sure, doctor.
Okay,
B.
others, can you please explain to Osama why it's B?
Self-kinase form at the position 4.
By position.
Yeah, I go to the
It's
slide.
the OH bar opposition to the alcohol.
No,
Okay, go to the
The
slide
fourth
where
position.
it's.
Well, it only works in the 4th position.
Okay, now look at this. Osama,
Yes.
can you see the changes? What is happening here? The sulfokinase will convert the OH that is on the para position, second OH, into HSO4. Okay, is it clear now?
Clear.
Okay. So when it is methyl COMT, it will convert which hydroxyl? The first hydroxyl, right? The first hydroxyl into what is the chemical changes happening? OCH3. Okay, so OH become OCH3. Only thing is the O-methyl transfer. That means a methyl group is adding. So same question. This similar way I can ask another question. Okay, I can just play around with the methoxy group here and there, and I can ask which of the following is an end product of the action of COMT. All right. So I hope with this you have understood the chapter. So we are going to have our next quiz on next class, that is on Wednesday, on the respiratory. It will be for 10 marks.
Can we make it on Monday, please?
No, we
Because
cannot make it Monday. It has to be on Wednesday. No chance. So
Why?
I tell you the
Because
slide
we have
numbers.
2 quizzes tomorrow.
Everybody
And
has
we
some
have already
quiz
2
the other
quizzes
day
on
about-
Tuesday.
So it has to be Tuesday. You have to study from slide number. I show you from which slide? From this slide. Okay, slide number 9. Okay, you don't have to study the beginning slide, 7 slides. So study from this slide. Can you see this slide? This slide, yeah. Until Benbuterol. That's all.
متى الكويز؟
Yeah. On Wednesday.
هاي
يعني كل الشعب عنده حركتين مش عارفة كيف يعمل.
So Wednesday for 10 marks.
Please make it on Monday.
Okay, if you have any other question, please, you can ask me before I finish the class. I still have 5 minutes time.
Doctor, so can we make it on Wednesday, but in the evening?
No, I cannot. First of all, you should not make such request. Okay, I mean it. I'm sorry to say this in a rude way, because it is you student who complained against the teacher who gave quizzes in the evening. And how could you ask? So
Okay, doctor, but we have a quiz tomorrow.
Didn't.
After tomorrow, we can't.
It's not you. On your behalf, some student representative, they complain and make them feel embarrassed in front of the whole university. So please do not make this request. Yeah.
They represent themselves.
But your class representative was also present. He did not defend. You know, the many class representatives were present. When somebody says nonsense, the people who were present from the class, they did not defend. When you don't defend, it means that you agree. What you say.
Can we make a vote?
No.
Doctor, please, we have.
You also. People have complained many things against me, so I don't give all these extra chances. So your quiz is on Wednesday.
Doctor, but
it's not fair.
Yeah.
I'm really sorry.
Because we used to discuss the date of the quizzes in our lectures.
These are the, okay, let me tell you, these are the things that I used to do extra as a benefit. Many extra thing I did, but students from your class, they complain that I am doing extra time, I am taking extra time. So you never bother to complain